So this is just an update from the Oklahoma team. Caleb Paisley, he is here with me. He will also present his field data. So he's a graduate student working on this project toward his PhD. And we have planted the plot in Bixby Research Station, Oklahoma State University, OSU. These are the 19 rows. We almost put 2,200 rose plots in 2 different plantations. And around 2,000, I think, survived. And it looks much better now. You can see all those flower, big canopy of the plants. And last year, Francisco and I, we inoculated with Rose Rosetta some of the plants by clip inoculation. And we see some symptoms. These are the field observation that Caleb, this is Caleb. This is my other student. She's not on this project, but she was visiting with us. So you can see different sort of typical virus-like symptoms. And then also some symptom like this. So Caleb is going to present his field observation now, what he did so far on the data that he collected.
Hello. Let's get over here. Okay. So yeah, these are just our observations from the Bixby research field. So I'll go over those briefly. So we've collected some data over time. We've just been collecting 200 randomly sampled plants just from our field. And you can see on the top right, the incidence. We're looking at both virus-like symptoms as well as fungal leaf spot. And so you can see the incidence over time for both the virus-like symptoms and the fungal leaf spot there. And then on the bottom right, you can just see you can see the different plants and the way that we're scoring them, actually very much like the presentation on the website, how we're scoring the symptoms that we're noting. So this is just based on observations in this case. And so just to briefly summarize it, we've noted just an average incidence from month to month. For the virus disease, it's 14.8%, and fungal disease as 52.3%. So we're seeing a lot of fungal leaf spot, particularly in this Oklahoma research field from what we're observing. And so just to briefly show a little bit more of the data here, for November, just to give you an idea, we observed 17% incidence in November here. And so you can just see the plots for those that are showing virus-like symptoms as well as the severity scores associated with those plants. We've also introduced 2 rose rosette infected positive plants that we've confirmed in the lab. We introduced those to the field. We found those actually at local grocery stores and nurseries, and so we were able to introduce 2 infected plants to the field. And from what we last have observed, they're establishing nicely. Just to overview of the fungal cultures that we're getting. This is just to give an idea of surface sterilization from the leaf and the associated cultures that we're getting from the leaves here. And then some of the leaf spots that we've noted just with the culture, so you can see that. And then same thing here as well. So just a lot of different images of the cultures we're seeing. So just to conclude that, we have screened 75 samples by RT-PCR to confirm if there's rose rosette occurring there or not. We haven't had any positive results for rose rosette by RT-PCR, and based on observations, there's not been any indication of rose rosette based on the symptoms observed. We've also detected rose cryptic virus in a number of samples by RT-PCR, and then again, we planted those 2 rose rosette infected roses that were confirmed by RT-PCR in the research field. And we've screened a number of cultures as well by ITS and EF1 primers to see if we're getting diplocarpin rosé. And thus far, we've only gotten alternaria based on the PCR results and subsequent Sanger sequencing. So that would be our conclusions with this.
All right. Any questions for Akhtar or Caleb? I would like to show another slide.
Okay, go ahead.
Yeah, so Caleb, if you can close that one. Ready?
Stop sharing. Stop sharing.
Okay.
I'm trying to. Is this? Sure. Can you see it?
Well, you can see it, Francisco. Go ahead.
Okay. Yeah, I just wanted to show probably some of the attendants are not familiar to where the lot is in the OSU Mingo Valley Research Station in Bixby, and there is a quite uniform lot of soil. So we try to minimize the effect of soil in this lot. So it's quite uniform. And the other thing is a comment regarding the incidence of rose rosette, which is going down, and it's very difficult to find plants infected with rose rosette. I found 3 symptomatic plants in the Botanic Garden, and I'm negotiating with them to transplant these 3 plants in the spring. It is because, I mean, the high mortality caused by rose rosette in rose plants has been very high during the last 2 years. It's quite difficult to find infected plants now. And then from there, the need that we have from the inoculum. And we are not allowed to bring rose rosette from other states. So that's complicating the situation. So anyway, we'll be looking forward to have a strong inoculum or spotting plants from where we can go and collect next spring, and then creating a heavy or a heavier pressure of inoculum in the field next spring.
Any questions for Francisco or Akhtar or Caleb?
More of a suggestion than a question. We have had success in keeping rose rosette infected plants for multiple years. And what we do then is plant additional healthy plants close to them and let those become infected, so that even when the originals eventually die, and it's usually winter kill, then we have a fresh batch of plants coming on, showing nice infections. So, and it would be good to put some screening or something around those infected plants in the winter to reduce the probability of winter kill.
Yeah.
Yep.
Yeah. Well, one of the things that we were planning to do is inoculating plants in the glass house in the winter. Problem is that then there are some other problems with mites usually in the glass house in winter time. So, and we cannot apply miticides.
Well, I can sympathize with issues with the mites in the greenhouses, but you can also easily infect the plants in the greenhouse by grafting.